Hello, everyone. Welcome to another webinar of the Amphibian Specialist Group. We are going to start the webinar shortly, and if you have any questions, you can write in the chat or ask yourselves at the end. So today we have with us Dasi Ong. Apart from being part of the Amphibian Specialist Group, Dasi is a PhD candidate at the Analytical Biochemistry Research Center in Malaysia. And today she is going to talk with us about how the biochemistry of the skin secretions in frogs has to do with their ecology. So thank you so much, Dasi, and the floor is yours.
Thank you, Marisa, for the introduction, and thank you, Amphibian Specialist Group, for the opportunity to share my PhD work. Hi, everyone. I'm Dasi Ong. Today, I'm presenting my research on understanding frog ecology through their skin secretion biochemistry. So, for the introduction, beyond just frogs, what their skin secretion can tell us about the environment. Yeah, we all know where there's frog, the environment is healthy. But here's the big question. What happened inside the frog when the environmental change? Are they actually happy or they just stressed and not telling us about the... So frog skin is a nature's biolab. Their skin isn't just slimy, it's sensitive. Other functions of the skin is actually to absorb water and chemical. It secretes biochemicals such as peptides, alkaloids, and metabolites, and also responds rapidly to stresses. So ecology and biochemistry, what is it? Unlike humans, organisms, plants, and animals use biochemistry to communicate and interact with each other and the environment. So their function is actually to interact and adaptation, defense, communication, and also a tool for survival, defense. My biochemical chosen is actually secondary metabolite. Yes, why secondary metabolite? So it's actually functions as defense and also communication. And peptides is for survival tool such as antifreeze proteins found in polar bear for cold and defense against antimicrobial infections. And also, communication via pheromones. So this is my species of choice, my favorite part, introduction of the frogs. So it's Hylarana erythrae, green paddy frog. It's from the family of Ranidae. Through frogs, it is very easy to identify. It's green with a two distinct white dorsal line on its back. It's a habitat generalist and very common in Southeast Asia, especially in Malaysia. In IUCN status, it is categorized as least concern. Only a few studies had been done for protein and peptides for biomedical discoveries, but in metabolite and natural peptide for ecological insights, there's none. So fieldwork were done in three main places, Langkawi Island, Penang Island, and also Kulim mainland. Permissions of this fieldwork were actually granted by the Wildlife Department of Malaysia, Palihilitan, and also USM Animal Ethics Committee. This is all my sampling size. All of them are pond size. You can see in the photos there. Some of them is just beside the road, urban, rural. Some of them is in front of someone's house or resort. One of the sites is actually in Tropical Spice Garden and also a protected place. And even one of the sites is in our university. So sample were collected 5 adult frogs per each site, hand caught. For frogs, unfortunately, there's no bait, so we had to hand caught. Fortunately for me, there's a aquatic net to assist my catching. Yeah, that's me inside a water catching erythrae. Yeah, the challenges is kids escape. They escape because they jump so fast for this frog especially. So the frogs were bring to the lab alive and well, and also still station. Measurements were recorded. And we actually released the frogs after the skin secretion collection. Yeah. Skin secretion collection were by physical shaking. Yeah, that's me with the frog in the plastic. Do anyone of you guys watch Nemo? There's a scene with a girl named Darcy. Mine Dasi, different. Yeah. She put a fish and she shake the fish until dead. But in my case, I only shake the frog for 2 minutes and rest them for 3 minutes. yeah, fortunately for all of my frogs, they are all alive and well and were released the next day. The skin secretion were then fish dried. Now, this is my general workflow. I won't go in depth in this part. So fieldwork were done, then sample extraction were done for metabolites and peptides from the skin secretion. Chemical analysis were done using liquid chromatography mass spectrometry, data identification using software such as MZMine, PICS, functional bioinformatics using metaboanalysis, KEGG, gene ontology, and so on, biomarker identification for potential biomarker compound. Then lastly, I'll talking about environmental stressor ID using general linear modeling to correlate the compounds to the stressors such as environment, temperature, and pollution. So my first research question is, what are the ecological and physiological function of this compound? For metabolites, we found that the amino acid and peptides are the most found in skin secretion, and these functions as antimicrobial peptides for the frog's defense. Second is alkaloids by 13% as a toxins for predator deterrence, and also fatty acid, which promotes skin repair, boosts immunity, and provides antimicrobial support. So for the pathway, I won't explain too much technical stuff. So basically, its function as protection, regeneration, antioxidant, antimicrobial peptides, and also homeostasis, osmotolerance of the frog. Wait, okay. So for biological process of peptides, we actually identified, you can see that we identified T cell differentiation in thymus, adaptive immune response, and also B cell differentiation, which is all related to immune system. And the tests were from nucleus, extracellular region, makes sense, it's the skin secretion itself, and cilium. For molecular function, it actually flagged the most in immune system process. So in general, the metabolite and peptides identified were environmental cues in frog skin secretion, which functions as stress, homeostasis, signaling of the frog, defense and protection, and also immunity and pathogen control of the frog, which is important for survival and adaptation and ecological niche of the frog itself. So let's go to my second research question. Which metabolite and natural peptide actually respond to habitat variation and can potentially act as environmental biomarkers? So for this, we use principal component analysis by biomedical compound mapped against all the sampling sites. For metabolites, you can see there's a clear pattern of separation between group 1 here and group 2 here. All of the colors correspond to different sites and replication of samples. Same pattern you can see here in peptides. There's two different group also, group 1 here and group 2 here. So for the candidates of potential biomarker, we actually use PLSDA by plot. So yeah, group 1 is characterized by this compound. And also this is group 2 for metabolites. Same thing we can see, same pattern for peptides. This is group 1 and group 2. So you might ask, why are these different? What are the similarity of these biomarkers? So from the list of potential biomarkers in different grouping of biomarker compound, we actually explore the physical-chemical properties of them, which it actually describes the substance chemical makeup, what it's made of, and how it behaves chemically behavior. So how it looks, reacts, and moves, such as major class of the metabolite, pH, hydrophobicity, charge of the peptides, and so on. So from there, we found out that group 1 metabolites actually associated with stable physiology and good metabolic health in frogs, which lets them sustain energy and maintains tissue upkeep. For peptides, is actually chronic for protection and host safety. So for group 2, is actually dominated affected by metabolites linked to defense and immune activation, such as exposure to with the environmental stress. For peptide, is actually peptides are more potent for antimicrobial defense. So conclusion here, we can say that frogs in group 1 habitat and actually provides a baseline protection for the frogs, while group 2 activates a more powerful active defense under environmental pressure. So this here is the final question that I'm answering. Which ecological conditions are the key drivers for the biochemical composition of Hirana erythrae skin secretion? For that, we actually assess the stress and physiological responses towards the ecology via biochemical composition, including metabolic categories, numbers of metabolite and peptides identified for each frog, eh, each site of frogs, and also a relative abundance of these compounds, and also concentration and yield of those compounds. Next, we actually map it using general linear modeling, using ecological parameters, which include 15 habitat quality assessment, including pond size. You can see there it's too small, but yeah, in general, it's 15 habitat quality assessment and also 15 water quality assessment, including pH, dissolved oxygen, sins, phosphate, total dissolved solids, and so on. So this is the general AIC of the general linear modeling. We can see that ecological parameters were based on the red box here are influential towards overall frog responses, and actually particularly in the green box here. I'll explain about it later in the next slide. So basically, all of this give stress to the frog, but the green box there actually give more impact to the frog. So what they say is ecological health can be monitored using this biochemical compound in frog skin. The strongest predictor, which is the green box earlier, showed that water clarity, arsenic concentration, aquatic vegetation, and pond size actually causes this frog stress. And site habitat assessment shows that low habitat sites such as tropical spice garden have a clear water, high dissolved oxygen, and also natural pond structure. This biochemical compound demonstrate a value as early warning biomarkers. So, in the term of my frogs, Helena atri, for them to be happy, so they need minimal water clarity, large pond size, presence of aquatic vegetation, and low concentration of arsenic level. Why? Because they're selfish. So they want spaces for themselves. They want to avoid competition and also hide from their predators. Just kidding. So this kind of study can also be like conservation. So the first one is proactive amphibian conservation by identifying the stress signal in rich ecology from being stressed before the population actually decline. Secondly is for freshwater monitoring system. Is research actually provide a molecular level monitoring tool and also a rapid integrative assessment, not just the ecological side of them, but we also integrate the organism, so the animal side of them. And the third one is disease ecology insight. We actually can monitor Rana virus and BD virus that understand these pathogens' susceptibility, which causes global amphibian threats. Lastly, biological indicator and environmental health. We know that if the frogs are stressed out, so the environment is dirty, and it actually can tell, actually will impact humans also. So some of the, all the research have challenges, limitation, and future application. Mine is escape. They jump so fast. Actually, in my case, and yeah. So other environmental factors influencing secretion were not done completely. Yeah, we only assess 15 habitat and 15 water quality. We might want to include frog diet pre-composition in the future and also skin microbiome. Limitation is this study is actually targeted on one species specific and method dependent, and data represent a snapshot and a long not a long term since actually just sample in dry season and a high cost and limitation functional validation is needed for the biomarkers. So future application is biomarker will need to be isolated, verified for the frog's health, habitat quality, and conservation purpose. One health links to ecosystem and human health need to be validated. Then source of bioactive compound for therapeutic use is actually a different topic for another day. So some of the publication from this research is this one and this one, and yeah, more to come soon. I would like to acknowledge my supervisors, supervisor, mentors, field assistants that helped me in catching frogs and support throughout out my journey. Fundings are from Nagao Nature Environmental Funds, Japan, and also Ministry of Higher Education, Malaysia. Sampling permission were directly obtained from authority of the property. And Nemo use and collection is actually permitted by Animal Ethics University Sains Malaysia and also Wildlife Department of Malaysia. Thank you for listening. Any questions?
Thank you so much, Dasi. Such an interesting topic, and congratulations on all your work that you are doing. As
Thank
I said
you.
before, anyone who has questions can now write them in the chat or just raise your hand and ask yourselves. Well, to start, I just have a curiosity. Could you do, well, I don't understand these kind of topics, but could you do swabs in the field instead of collecting the frogs to the lab?
Sure, if you have enough sample, you can do that.
Okay.
Because skin secretion, if using my kind of method, we actually have too little sample for it. So if swabbing, I would think, yeah, better change method for swabbing to other things because you
Yeah.
might get lesser biochemical compound with
Yeah, yeah.
swabbing.
Yeah, okay,
Yeah,
I understand. Okay, thank you.
you're welcome.
And also in terms of conservation, uh, would you plan or are you working with any kind of entities? Where did you collect the samples to try to improve some environments based on your findings? Or could you just
For
reach someone to suggest these are what we found and we could improve or not?
For now, no, not yet, because this is a tryout on the model of research itself. So I actually wanted to approach some NGOs and share about this finding and hopefully improve some of the habitats. Oh, Tropical Spice Garden actually approached me for the findings and talk about how their pond is actually clean
Okay.
and with a lot of rocks.
We have here a question in the chat. So, based on your findings, are the frogs happy?
Uh, s- the frogs in clean places are happy, but not all of them.
Yeah, of course. Yeah, that's why improving habitat would be good for them. Another question, does secretion be an indicator if the frog is poisonous?
Wait, let me read the chat. So does question be an indicator? Yes, you can actually also screen for the compound that is poisonous in the skin secretion, such as alkaloids and toxins.
Okay. But the ones that you were studying were poisonous or not?
In Malaysia, no,
No, yeah.
not that poisonous to human, only to small animals.
Yeah, just the toxins for defense, right? On predation.
Yeah. Yep.
Does anyone have any other? Oh, there is another one. What factors did you consider in choosing the sampling sites? Why choose urban areas? Or, if possible, why not compare the skin secretion of frogs from urban areas versus forest areas?
Oh, this is an interesting question. So why urban areas? Just because there's a lot of frogs. That's why I choose the sampling site. That's one. Actually, we did do forested areas, such as in the tropical spice garden. It's semi-forested areas and it is protected. Yeah, and some of the sites we actually can't find that much Eritrea. I did one survey in a forested area, but unfortunately it's not a pond. Oh, I forget to mention all of my survey sites are pond areas. That's why. So for forested areas, we can't find those kind. And not a lot of Eritrea in forested areas. That's one more thing. Since Harana Eritrea is more common in urban areas.
Okay. But probably you would find different results, no?
Yes, you might find different results with different habitat.
Yeah, maybe it's a question to test in the future.
Yeah, I need to do that also. Thank you for that.
Okay, another question. Good day. May I ask you, did you observe any differences between frogs caught in polluted water compared to clean?
Do you mean by in term of the size of the frog or the physical of the frog?
Mm, not sure. Maybe you can ask both. You can answer both.
For yeah, for clean areas, they are actually lesser frog and harder to catch. For not polluted, more to slightly dirty waters. Yeah, and there's actually more Hyena erythrae to be caught, and it's more easy. That's what I find.
Yeah.
In terms of physical size, no difference. Yeah, but in urban areas, yeah, frogs are tend to be more injured by the grass cutting, so they injure their legs. Sometimes they lose one of their limbs. Yeah.
Okay. So I think there is a question here, but I'm not sure if you can answer. That is, comparing the biochemistry composition, what are the implications of the skin secretions collected between the urban versus the forested area? But you didn't test that, I think.
I did one of
Ah,
the
okay.
sites.
Okay,
Yeah.
okay.
So yeah, I got actually got an answer for that. So in cleaner area, frog skin secretion tend to have more lipid instead of urban areas. They tend to have more antimicrobial peptide for protection purpose. Does that answer your question, Luis?
I think so. Well, another question. Hello, thank you for a good presentation. My question is, how species-specific are these skin secretions biomarkers? Can some biochemical indicators be applied across different amphibian taxa, or do they need to be developed and validated separately for each species?
Oh, yeah, they need to be developed and validated separately for each species since different species secrete different chemical compounds.
Okay. What do you think would happen if a toad were included in your sample? Sometimes some toads are better adapted to urban areas.
I'm actually thinking of doing that for my next project, taking toads instead of frogs only. So compare between the frogs and the toads conversation. So I would think, yeah, quite the same as Eritrea since they are more to urban species.
Yeah, maybe you can answer that question in next year or in a few years.
Funny.
Another one. Could you elaborate what you meant by water clarity is the turbidity levels?
It's actually the same. One is by physical observation, water clarity. Another one is by lab testing, turbidity.
So you use like different scales.
Different approach. One is by looking at it. Another one is by chemical testing.
Okay. Okay. Excellent research, Dasi. Are there consideration of age and sex in frog sampling? If that is, will they have an effect?
For me, unfortunately, I only take adult frogs without considering the sex and age. Oh, the age is actually adult as a general. Not I didn't consider the sex for frog sampling. If I do, I would think there's slight difference, but not much since they are the same species. Yeah, since I can't consider sex for something just because even catching five per something sex is hard. If I consider male and female, it would take me at least twice the hassle to catch frogs in the field.
Yep. So why choose this specific species? Why not another species secretion?
Because it's very common species in Malaysia and it's easy to catch and find in urban areas in Malaysia here. If I choose a rarer species, yeah, it will be a good research, but when am I finishing my PhD? So maybe for future works or a long-term work, I would choose more threatened species so we can know their conservation value. That's why.
Yeah.
Thank you for that.
Here you have a compliment. That was a great
Aw,
presentation. And then
thank
another
you.
question. How can the biochemical composition of frog skin secretions provide ecological insights that are not evident from gene expression and analysis of skin tissue alone?
This is a very good question. So, biochemical composition such as metabolite and peptides is actually respond to quick environmental change, unlike gene expression and tissue analysis alone. That's why we choose metabolite and peptides for analysis. So it actually responds faster to environmental change.
Okay. So here in the chat, this was the last question. I don't know if you have any others. You can also raise your hand if you have.
Or you can also email me.
Exactly. Not sure. Dazi, you can write your email in the chat, maybe.
Sure.
So
There.
if there aren't any other questions, we can finish perhaps. Well, thank you so much for everyone that attends today, and I hope to see you in the next webinar. And thank you so much, Dazi, for presenting us your work today.
Thank you.
So bye bye, everyone.
Bye.